we were on this part, and at the very top of it, we were looking at acetylating histones. So I could add acetyl groups to histones. That kind of like neutralized the positives on the histones. They'd let go of DNA. Now genes were more likely on. And then the reverse was true. DNA methylation, we've been spending time on like 1, 2, 3, 4 right here. We've done like 1, 2, 3, I believe. I'll double check my little post-it note. But I think we've done 1, 2, 3, and we need 4. But that was, I can add methyl groups to promoters, and if I add a methyl group to promoters, that tends to turn genes off. They're like blocking RNA polymerase from landing there. So things that we saw that methylation patterns matter with like calico cats. So if I have DNA and I like change methyl patterns, then now that cat might have this phenotype. That fur is orange. Or it might have this other phenotype. That fur is black. Next up, we saw that methylation patterns can switch throughout life due to life experiences. That's when like mama mice would lick her pups and methyl groups would fall off and they'd be like well-adjusted adults as opposed to anxious adults. Those are yep. So the third one was we looked at Angelman and Prader-Willi. What was the deal with Angelman and Prader-Willi? That one's pretty tough to describe. Like, what was the significance of that? Yeah, good. A mutation from different parents can cause a different disease. So if I have a mutation on my chromosome 15, the next question would be like, hey, was that mutation, if it was this particular one, a deletion of this little bitty segment on chromosome 15, it's like, hey, was that deletion from your mom's chromosome that you got from the egg or from dad's chromosome, the sperm? And you're like, why does it matter? Different disease. Not all genes work this way. Eye color, for example, big B, little b. If I'm big B, little b, I'm going to have brown eyes. It doesn't matter if the capital B came from mom or from dad. But if that gene was marked this way, all of a sudden it would. Like, oh, you're big B, little b? What's your eye color? And it's like, well, where did the big B come from, mom or dad? And you'd have a different phenotype. So because this happens on chromosome 15, dealing with Angelman and Prader-Willi syndrome, we assume that it happens other places too. So if you're interested in it, they call it genetic imprinting. So not all genes do this. Not all genes, again, not all genes matter if you got it from your mom or your dad, but some do. So we assume that this is possible in like other places too. It's not just this one-off thing. We assume that there are places on my chromosome 2s that if I got the gene from mom or I got it from I got the gene from mom or the gene from dad, my body would like treat it differently. So that's kind of strange. All right, the last one, this one's new. I can mess with methyl patterns in a lab. So I can do experiments and like see, hey, what happens if I add a methyl group and turn that gene off? Or that gene's off. What if I remove the methyl groups and allow it to be turned on? Yeah. The feast or famine we'll skip. This happens to be like the original studies where they decided that like things that happen in life can get passed on to future generations and kids. I don't like it. Like, all scientists agree that it's a solid study, but when I read it, I don't totally understand how it's a solid study. It seems like it has holes in it. They're correct. If a bunch of dudes, biologists from Harvard and MIT are like, oh no, this is solid, and I'm like, but I have questions, they're just like, shut up. So it's my own lack of understanding. But basically the quick version is they went back, and I don't remember what group it was, but there are certain people in the world that keep like really, really tight records of like genealogy. For example, in the US, if you're Mormon, part of being Mormon is you have to be, I'm not Mormon, so if I mess this up, I'm doing my best. But if I'm Mormon, like who I'm descended from matters. Like my bloodline matters if I'm Mormon. And so like Mormons are really good at like genealogy of like, they know who their great-great-great-great-grandparents were, who they're, like they keep track of genealogy for religious reasons. So there was some group, and it wasn't for religious reasons. It was in like Sweden or some place like that. And they kept like really, really tight records. And what they found was they could predict how long kids were going to live, like their life expectancy, based off of whether their grandparents survived a famine or they were around during a feast. So like Was food plentiful, or was there like some sort of natural disaster and like everybody was starving? And the difference in life expectancy was significant. It was like 8 or 9 years. And they could predict, like statistically significant, predict like, hey, here's all these people that live this long. Who do you think their grandparents were? And they could predict like, oh, their grandparents lived through the feasting times, or their grandparents lived through the famine times. By the way, the famine times were the ones that had kids that survived the longest. So they survived. So the grandparents that had a cush life, their grandkids died, again, like almost a decade earlier. Now, if you start asking questions, that's where I'm like, hey, I'm trusting the other biologists. My questions are like, well, aren't there like all these other factors? And they're like, no, they controlled for all these factors here. Temperature, like, you know what I mean? Like, I'm like, I need more details. But supposedly, like, they went through and they controlled for all the other factors, where it's like, what about temperature? Were people just freezing to death? No, because in the summer of this and the autumn of this. And so, like, people that know this stuff well, they're like, no, it's like, it's really good evidence. That's when they first started thinking that, like, you could pass on methyl patterns to future generations. Now we have really good experiments, like the mom licking the pups, where I can, like, really show you in isolation that, like, methyl patterns matter. So now we have better experiments that are controlled in a lab. Does that sort of make sense? So this is the one that was just sort of out in the wild that got all this started. But now I'm going to prove it again in a lab. So number 4 is more solid evidence. If I'm a student and I'm like, I'm not sure if I believe you or not, Cotton, after number 4, it's going to be tough to not believe me. The feast and famine, if I told you that story, you'd be like, yeah, I don't know. So number 4, methylation patterns can be manipulated in a lab. Okay, so think back. Do you guys remember this picture? Yeah, what was that mouse on the left called? It had a weird name. Yeah, the agouti mouse, and it's made an appearance on the ceiling tiles. So that's an agouti mouse. And we learned about it with pleiotropy. Remind me, what was pleiotropy again? Like, that's why we saw this picture back in the day. Huh? Yeah, one gene can be the cause of multiple phenotypes. So I could have one gene and it can determine my fur color phenotype, but also my hunger and maybe whether or not I'm at great risk for like liver cancer or something. So it's when one gene can control many different phenotypes. In humans, there's one that I know of where there's a potassium channel that we use in our kidneys. So if it's bad, my kidneys are messed up, but we use the same potassium channels at the cellular level in our ears to hear. So if that potassium channel has like a bad genetic code, my kidneys are messed up, which is like a completely different body system from my nervous system, and my hearing's also messed up. So that's pleiotropy. So that is true. And pleiotropy happens because like multiple genes are involved. So pleiotropy, this example can happen for 2 reasons. This guy, it's like a zoo out there. This animal right here, it could have a deletion. And so it's got a screw up in the gene, and so its fur color's messed up, its appetite's messed up, all these things are messed up, and then this one's fine. However, sometimes in these pictures, they're actually twins. So those are twins, identical twins. And you're like, no, they're not. And I'm like, yes, they are. And you're like, no, they're not. And I'm like, yes, they are. And we do that for like a minute. And then you're like, well, how are they twins? Here's how. So I've got this mouse, and we'll just call this typical. So this is the typical mouse, and then this is the agouti one, so he's going to have all these health problems. I've got the agouti gene. That agouti gene is supposed to be methylated, which means it would be off. If the agouti gene is on, I'm like this. If my agouti genes are off, I look like this. Just like pretend that there's me and pretend I have an identical twin brother. I don't. But pretend that I'm big B, little b for eye color. So I'd have brown eyes. And my brother is big B, little b for eye color, because we're twins. So he should have brown eyes. But what if his capital B gene was silenced? All of them. In every cell. Wouldn't he have blue eyes? I'm big B, little b. He's big B, little b. But if his capital B gene is off, he'd have blue eyes, right? So that's what's happening here. They have identical A, T, G, and C because they're identical twins. This one, some genes are methylated. That's why it's like this. This one, those genes are not, but they're twins. All right. BPA. Have you guys ever heard of BPA? All right, this is one of the common kind of contaminants that we're worried about that could be in our food, our water bottles, our plastic, out in the environment. It's bad for other animals and stuff. The tough thing with like environmental stuff is it sure seems like every week.
Teachers and students, please pardon this interruption.
Okay.
Teachers, please hold students until further notice when you hear from me for this period. Please hold students, disregard the bell, and do not release them for this lunch. Thank you.
Possibilities. It's not lockdown, or they tell us to like lock our doors and stuff. So I don't know. Maybe they're running the drug dogs. I don't know. Anyway, we've got BPA. The problem with any sort of environmental toxin is like it seems like every week somebody's telling me another thing that's like in my food, in my water that I can't eat or drink. And at the end of the day, all of a sudden it's like I can't have meat, I can't have plants because they're covered in pesticides. I can't have the plants that have this. I can't have that. I can't have plastic. I can't have metal. Before you know, it's like I can't eat anything. Everything is unsafe. Then I read the study and it's like, hey, like as long as you're not eating like a pound of plastic a day, like it's okay if you use like a plastic fork. Just don't eat the fork. And if you eat the fork, just eat like one. Don't eat like a box of plastic forks and then you'll be okay. And you're like, you buttholes, why didn't you just tell me like, you know, it's almost like, watch out for water, you know, too much and you die. True, but like, I just won't drink too much. BPA. BPA has a recycling symbol of number 7. You know what that means? Not recyclable. So somebody decided, all right, number 1 has this in the plastic, number 2 has this in the plastic, 1, 2, 3, 4, 5, 6 means recyclable. Number 7 means can't be recycled. They didn't do this. Here's what I would have done. I would have kind of like had the symbol. And if I wanted to tell people like, hey, don't put this in your recycling, I'd have done like, I don't know, that, or the classic this. I wouldn't have done, oh, now people will know. So if you look at the bottom of styrofoam and it's got a 7 on it, don't put it in the trash. It's not recyclable because it has BPA in it. So when I look at this, again, if I'm at home and I'm recycling, I'm assuming this is what 99% of you guys do. You look, and you're like, can I recycle this? And you're like, oh yeah, it's got the little triangle. You aren't like, what number is it? No, that's a 2, so it's fine. But if that was a 7, I have to throw it in the regular trash. So 7 is not recyclable. Isn't that bizarre? That's dumb, right? Okay, so now the general public would have to know that like 7 is recyclable or not recyclable. So that's BPA. And you're like, all right, well, who cares if you have BPA? These are offsprings. They're identical twins. So we'll just say that they're brothers. They both came from the same moms. The moms were also clones. So the moms are genetically identical too. So if I'm looking at them, they look different, but they're twins and they came from the same mom. So, or the same, different moms, but the moms were twins. So his mom, she had access to regular food, and then we gave her however much BPA. And that's why her kids are messed up. The other mom had access to the same amount of food. We hit her with the same amount of BPA, but we also gave her folic acid, vitamin B12, betaine. I had to look up how you say it. That's how you say it. Instead of betaine, it's betaine. And so those mice turned out to be fine.
Pardon this interruption. You may release for second lunch now. Yeah. You may release for second lunch. Thank you.
I'm in my video class, and I've got like a glare on the screen. I fixed that. All right, so those guys are twins. And so what they found is basically if I give mom BPA and a regular diet, her kids turn out like this. If I give mom BPA, her regular diet, and like B vitamins, the kids turn out like this. So here's what we found out. BPA knocks off methyl groups. That's why I'm not supposed to have it in my plastics. So if I have a mouse that's like this, and I consume enough BPA, I don't know how much as a human I have to consume, it'll knock off those methyl groups. And then I become this mouse. You're like, all right, what is BPA commonly in? Baby bottles. That's unfortunate. That's when I'd be most susceptible, right? Like, let's say that you give me this much BPA. Well, if I weigh like 160 pounds, that's a small percentage of my body weight. Let's say you give that same amount to an 8-pound kid. Well, now that's the equivalent of 20 times the concentration because that kid's 20 times smaller, right? You know, if this much Tylenol is toxic to an adult, if you give that to a 8-pound kid, that's like 20 times the toxic level because that's an 8-pound kid. So it's not supposed to be in baby bottles anymore. When you guys are old enough, if you buy baby bottles, buy them from a store. Don't buy them from Amazon, from somewhere overseas. Buy them from here. They're more expensive at Target. Yeah, but they don't have BPA in them. If I buy them from some random place off the internet, I don't know if they have BPA or not. So BPA knocks off the methyl groups. You're like, all right, but what about the kids of moms who got folic acid supplements, vitamins, we'll just say. Let me show you what beta-ene looks like. So first off, what was the chemical formula for methyl? Like the chemical formula for water is H2O. What was the chemical formula for a methyl group? CH3. And the problem, what was it that BPA was doing to me that was causing harm? Yeah, it was knocking off the methyl groups. Here's what beta-ene looks like. Oops. And like a bunch of other files. So here's what beta-ene looks like. CH3, CH3, CH3. So what is beta-ene and folic acid and B vitamins doing? Adding the methyl groups back on. So why am I supposed to make sure that I get enough vitamin B6, vitamin B12, and folic acid? So that I can turn genes off. That's why I need vitamin B12. It helps me turn genes off. Who gives a crap if I'm running low on, I need enough vitamin C. Why? So I don't get scurvy. So I'll eat an orange. If I need to turn genes off, I need methyl groups. So let's say in my body, it's time to turn genes off. I've got to turn off these 5,000 genes. I need to go into the shelf of Allen Cotton and have thousands of methyl groups to start like turning off all these genes. What if I don't have the methyl groups? I can't turn them off. So if I'm running low on B12 vitamins, or I'm running low on folic acid, or I'm running low on things that provide methyl groups, it's like, what are the symptoms? It's like, well, what cells are running low? If it's in my brain cells and they're running low, then that means there are some genes that are supposed to be getting turned off, and they're not. So what's the result? Well, which genes got turned off, and which one didn't get turned off? Isn't that crazy, though? Like, I don't know. I find it interesting when like, oh, now I know why I need folic acid. That provides methyl groups so that I can turn genes off when necessary. Yeah. Folic acid is B6, so vitamin B6. Folic acid is the only legal difference between vitamins and prenatal vitamins. So one day you females, you might be trying to have kids and you won't take vitamins, you'll take prenatal vitamins. And you're like, what's the difference between a Centrum and a prenatal Centrum? You get a double dose of folic acid. That's it. That's the only difference. So now there are plenty of methyl groups so that as your baby is developing, genes can turn off when they are supposed to turn off. So we double the amount of folic acid. When we didn't have adequate folic acid, the most common disorder is spina bifida. That's when your spinal cord grows outside of your body. And if your spinal cord is damaged, you're paralyzed. And damaging your spinal cord is about as easy as damaging like Velveeta cheese. So like if I have spina bifida bad enough and you just go up to my spine and you go like that, I'm paralyzed from there down. Again, like damaging brain and spine is about as hard as like with your finger damaging like room temperature Velveeta cheese. Like if I have room temperature Velveeta cheese and I just kind of go like thunk, I like dented the corner. That's how easy it is to damage your brain. Some of you guys might get to dissect like brains and stuff like that. One day you might dissect it in a lab and like you could easily just take like the sheep brain and anatomy and you could just like with your finger, like it's that easy. I don't know how I'm still alive. How can I play sports, fight, fall down the stairs? How is my brain not just a pile of rubbish? I don't understand. Yeah.
Is there like a specific protein that knows where the methyl groups go after the DNA?
That's an excellent question. So, yes, but I don't know. I'm assuming so. Just based off of pure assumption, it has to be. So the question is now coming down to like, where do the methyl groups go, or how do they know to silence this gene but not that gene? One thing that we'll do a little bit later today, it won't address like methyl groups and silencing genes, but it is going to be like, how does a bacteria cell know to turn genes on and off with just chemistry? So by the end of class, we'll have something that you could kind of envision as a solution on a simpler scale, but I don't have a good answer for you. Yeah. Other questions? Isn't that weird? That's why BPA is not legal to put in plastics anymore. By the way, plastics that are likely to have it are moldable plastics. So if you just have like a solid sheet of plastic, like pretend that this door was plexiglass, that's just like a solid sheet and it's really, really hard, that's unlikely to be BPA. BPA is in molded plastics, things with shape, curvy plastics like this, and water bottles that we drink out of, and baby bottles. So water bottles and baby bottles were what we were concerned about. Yeah.
When was it banned?
That I don't know. I'd have to look it up. But I would think it should be pretty straightforward on Google, like when was BPA banned from plastics or whatever. And in the U.S., it might have been banned in this year. And then in the U.K., it was banned in this year. And in California, it was banned like 70 years ago or whatever. Have you ever bought anything and it says California Proposition 65? May contain things that could cause mutations, blah, 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 blah. So there's crap that like you can't make in California that you can sell everywhere else in the United States. So they have different laws. So Dr. Jurtle, that was the one that did these experiments. He could even scale them down. So he'd be like, all right, all their mamas, these are all twins, all their mamas, I gave them all food, I gave them all BPA. These mamas, I gave this much of B vitamins. These I gave this much. 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 So he could even scale it down. It's not just like you're an agouti mouse or you're not. It's like I give you some methyl groups, so you're sort of agouti. So it wasn't just yes or no. It was like everything in between. So we know that methyl groups are like the cause of this. Pretty interesting. We can go in a lab. I don't know how we know where the methyl groups go. I've got a hunch, but I don't know for sure. And we can change, remember how we talked about those mama mice and making like anxious kids? We can make the adult mice anxious or not by adding and removing methyl groups. So we can change their personalities by adding and removing methyl groups. So the reason that we think like personalities are based partly on adding and removing methyl groups is when we add and remove methyl groups, we change your personality. So the reason that I think this switch controls that is because every time I hit down, it turns off, and every time I hit up, it turns on. So I'm pretty sure that like that switch is controlling that light because it does. Another example, turns out workers, drones, and queens are all the same. They're clones. I didn't know that. So the queen, I don't know how they decide, but they decide who's going to be the queen. And if they screw up and they make two queens, then the queens fight to the death, and the winner's like the queen bee. So they feed the one that's going to be the queen this stuff called royal jelly. Everybody else just eats honey. And they feed her royal jelly, and royal jelly goes and silences certain genes. It provides methyl groups to silence certain genes, and that causes her to be the queen. They don't get royal jelly, so they're not the queens. Like one of them's even sterile. I don't remember who it is. The drone has sex with the queen, or the worker has sex with the queen. I don't know. Somebody has sex with the queen. The drone does. Cool. And then the workers just, I don't know, I guess they're just building honeycombs or whatever. Do what? Yeah, but they're all genetically identical, but they're turning genes on to make them males versus females. So weird. So I find that interesting. This last one, this one's a little bit newer, so there's not, I will zoom in. So there's not as much evidence piled up, but as they pile up evidence, it's still supporting this idea. So I find this one fascinating. This is from Duke, a group out of Duke University. Modified experience, neuron DNA methylation is required for synaptic plasticity. Active DNA methylation and demethylation is therefore required for memory. So how do I remember stuff? I might be methylating and demethylating genes. So like right now, when I'm trying to remember, though, like if I add a methyl group to a promoter, what does that do? That turns a gene off. I might remember that by turning some genes off. Isn't that bizarre? So there's more to memory than just that, but they've done other studies. And again, it's not enough to be like purely in a textbook. This is one of the major ways that memory works. But like they're starting to pile up evidence to where in the future, this might be a thing in textbooks of like, how does memory work? Well, you do this and this nerve cell and this nerve cell, and they release neurotransmitters and all this. And at the level of DNA, we also like literally like turning genes on and turning genes off. And like, oh, you didn't turn that gene off. That's forgetting. Or, oh, you were supposed to turn that one on. That's forgetting. Isn't that bizarre? So now you could get down to like, why can't I remember stuff? It's like, well, you're running low on your B vitamins, so you couldn't turn the genes off. Like, it's fascinating if you have a chance to think about it and let it like sink in. Memory could be turning genes on and off too. And one of the reasons that I didn't do well and couldn't remember is I might have been running low on certain vitamins. Again, this is a little bit more fringe science, but it's not like just total BS science. All right. So the final conclusion on the turning genes on and turning genes off in humans is this. Do the right thing even if nobody is looking. Why? Well, your life choices and experiences can change the way that DNA is expressed in you. That makes sense. We kind of have evidence of that. What you do at age 13 can have lasting genetic effects that might be passed on to future kids, kind of like teenage binge drinking and moms licking their pups and stuff like that. Life events that you experience or impose on others can also change the way that DNA is expressed. So how I treat other people is turning genes on and off in them. So be nice. Now, if somebody's being a turd and they need to be like checked by society, check them. They need to learn how to behave. But like when somebody's having a rough day, cut them a little bit of slack. When you have a kid one day, I don't know, tell them what to do, but like, I don't know, just don't be too hard. All right. Any questions about the 1, 2, 3, 4 kind of DNA methylation stuff? I find this completely fascinating because we now have a chemical reason for like why I am the way that I am. Yeah. Yeah, that was just sort of like the evidence for creating neurotic mice is just kind of a quick little nod to sort of this. Like I can add and remove methyl tags, and I can switch back between like an anxious neurotic mouse and one that's just kind of like chill by turning genes on and off. Others? Makes sense that we'd have genes that say chill out, but it also makes sense that we'd have genes that say like, hey, you should be a little bit nervous right now. Otherwise, when I'm in a situation where like I should feel really, really nervous, it's like very dangerous, and I'm just like, everything's cool. No, it's not. Like, you're in a bad spot right now. You know, so there are times where like nervous is good, and there are times where like being able to chill is good, so I don't have a heart attack. So it makes sense that I'd have genes that say chill, be nervous, and just finding the right balance. Huh? So this is where they're currently working on with humans. Once they're grown, can we switch it back? I was reading about it, and I don't know that I ever got to the conclusion. So, but one of the arguments is, hey, there are some people, it's not proven, but there are some people that say part of the reason, if you look at like the average weight and like how many people in the United States have a weight problem now compared to 30 years ago, it's drastically different. You guys haven't grown up with it, but I have. The number of people that have diabetes and everything else is like, it really is drastically different. The world like looks different. Walmart looks different now than it used to, like drastically different as far as like health is concerned. And there are some people that are like, how much of that is because of plastics? How much of that is because we started drinking Ozarka? When I was born, there weren't, Ozarka wasn't a thing. There weren't water bottles. You didn't buy water. Water came out of a tap. If I went to the grocery store, I couldn't buy bottled water. There wasn't a market for it. Nobody paid $1 for water because it's free out of the fountain. So, right, like right now you pay more for bottled water than you do gasoline. Right, if I go to Quick Trip, isn't a bottle of water that's like 20-something ounces, isn't it like $1 or $2? And a gallon of gas is like $3. So we pay more for water. Like in that Quick Trip, by the bathroom, there's a fountain and it's free. So my generation, that's how we saw it. And so we're like, if somebody's like, hey, here's water for $1, like, no, nobody marketed it because they knew nobody would buy it. So at a store, you could get a 1-gallon jug if you're going camping or whatever, but like there was no like, I just want a small bottle of water. There wasn't Ozarka. There wasn't an aisle at the grocery store for water because nobody would buy it. Ask your parents. When they grew up, there was no Aquafina. There was no water bottle. There was a cup. And that's a faucet. That's why I had every year, I'll do this. I'll see what some of you guys give me. Like, I have students that ask, like, do you drink the water out of there? And I'm like, yeah. It's free water. It's fine. So anyway, we think BPA could potentially be the cause for some of our health issues because we've been drinking out of plastic water bottles with BPA, and our food's been in plastic things with BPA. Who knows? All right. We only need one last major thing so that we can be ready for our test next class. So if I go backwards, you get to Prokaryotic Gene Regulation. There's a page where you've got like these 3 pictures, just like one on top of another, like pow, pow, pow. These kind of 3 pictures. I think you go back to like a page. Might actually be just on it's on the left-hand page. So if you were on this is on the right-hand side of your paper, and then now you're on the left-hand side. All right, so here's a picture, and this picture is going to give you background information that's going to explain those 3 things. So this is kind of like the last big thing or the last difficult thing that we learned in this unit. All right, so I've got this right here, and let's say that I want something. Let's see, which order do I want to do this in? All right, let me ask you this. Is there stuff in my body that I want to pretty much always stay the same? Can you give me an example of something that should pretty much just remain steady no matter what, relating to my body? Like some stuff needs to change a lot. Like when I'm 5, I'm not producing a lot of testosterone, a lot of growth hormone or whatever. Then when I'm 15, all of a sudden I need to start, well, when I'm like 12, 13, I start producing a lot of testosterone. So there are times when it's like low testosterone, high testosterone. a whole lot of growth hormone, now I'm going through a growth spurt, or low growth hormone, now I'm not going through a growth spurt. So what kind of things would need to be steady pretty much all the time? Yeah. That's true. My genes, I don't want to like add and remove genes. What else? Your body temperature need to stay about the same all the time. If it's cold outside, you'll shiver because I want to keep my body temperature about 98, 99, right? If it's really, really warm, if it was 80 in here, I would sweat to keep my body temperature about 98, 99. If I chug a 2-liter Coke and my blood sugar is going to go through the roof, that'll kill me. So I have insulin that brings it down. If I haven't consumed any sugar in a long time, I have another hormone that called glucagon that brings it up. So I'm always like keeping my blood sugar like not too high, not too low. So there's a hormone that says less, another one that says more, less, more.
Doesn't your body purposely raise your body temperature when you have fever?
When I have a fever, true. So, but like, let's say I have a fever for a month, I need to keep my body temperature steady for a month. Now, I might choose to like raise it to 102, 103, 104 when I'm sick. That's true. But I need the ability to keep it steady. If I don't have a fever for a year, then I needed to keep it steady for like a year. But you're right. My body, I can choose to do something different. Yeah. So I need to keep something steady. Okay. So here's how I can keep something steady. Anytime that I want like something to remain pretty constant, I do what is called negative feedback. If there's negative feedback, what else do you think there probably also is? Positive feedback. So negative feedback, I'll do that one in red. Positive feedback, I'll do the graph for it in a second. I'm going to see if you guys can figure it out. Positive feedback, I'll do in blue. All right, so I got positive feedback and negative feedback. All right, cool. If I want to keep something steady, I probably control that through negative feedback. All right. So here's how this could work. I want to make this chemical called tryptophan. Why? Don't sweat it. It's an amino acid, so my body can make it. So let's say I need to pretend I have to have a steady amount of tryptophan. I have no idea if your body needs a steady amount or not. But for this example, we're going to assume that I do. So let's say that I always want 100 tryptophans. 100 tryptophans. Okay. How many enzymes does it look like, according to that diagram, how many enzymes does it take to make a tryptophan? How many different enzymes does it take to make a tryptophan? Yeah, it looks like 5. Looks like a moving assembly line. It looks like I've got worker number 1, worker number 2, 3, 4, 5. It looks like I basically take this chemical, whatever this is, and it looks like I give it to enzyme number 1, and he like runs this chemical reaction right here. Then I hand it off to enzyme number 2, and he like does this chemical change. And then like enzyme number 3 and 4 and 5 until like eventually you have that. Tryptophan. Cool. How many tryptophans am I pretending that I always want? 100. Okay. So how could my body know to have 100 tryptophans? How does it know how many I have? How does it know that it doesn't need to make more? Like, how is my body thinking this? How is it figuring it out? This is kind of what I said would relate to your question earlier. Negative feedback. Okay. So tryptophan is feeding back. What do you think it's saying to do? Do you think it's saying stop or go if it's negative feedback? Stop. So what does this process, this whole process, these 5 chemical reactions, what does this whole process eventually make? Tryptophan. What is going to tell the process to stop? Tryptophan. So I'm feeding backwards, and I'm saying stop. Negative. So this is negative feedback. Think of it this way. Pretend that one day you're a parent and you're nagging your kid. You're like, clean your room. Then you go and you check, it's still dirty. So what do you say? Clean your room. You go and you check an hour later, it's still dirty. So what do you say? Clean your room. What would you have to do as my son or daughter to get me to quit telling you, Clean your room. Yeah, you would show me a room that's clean. So I'm telling you, clean your room. What's going to make me stop? A clean room. I'm telling you, make tryptophan. What's going to tell me, hey, I'm done making tryptophan? Tryptophan. Hand me that orange. Hand me that orange. Hand me that orange. Hand me that orange. Once you hand me the orange, I quit asking you to hand me the orange. So give me that thing. Give me that thing. So whatever I'm asking is the product. That's what like stops the production of the product. Let me show you what I mean. Let's say I've got, I want to make 100 tryptophans. And so what I'll do is I'll have 100 enzyme number 1s or 100 gene number 3s. We'll go enzyme number 1. So I've got 100 enzyme number 1s. And I've got like infinity of each of these. So enzyme number 1 is where I control it. So again, once we understand this, then those pictures in your notes will make sense. So here's my enzyme. That's my active site. What was this alternate site called? It wasn't the active site. What was that one? Yeah, an allosteric site. And what can bind at an allosteric site? Not a substrate. Yeah, an inhibitor. So I could have an inhibitor turn that gene off, or I'm sorry, turn that enzyme off, right? If an inhibitor hits that enzyme, I turn it off. Okay, so what if I have 100 of these enzymes? All right, so cool. What if tryptophan turns them off? What if tryptophan feeds back and turns that enzyme off? Okay, so I've got 100 enzymes. Tryptophan can turn them off. So tryptophan can inhibit enzymes. So tryptophan is an inhibitor of this enzyme. I've got 100 of those enzymes. If I've made 75 tryptophans, what's the maximum number of enzymes I could turn off? I've made 75 tryptophans, so how many enzymes could possibly be off? 75. So wouldn't there still be 25 of them that are on? And what would they be making? Tryptophan. So I'm going to keep making tryptophan. Now I've made 90 tryptophans. In theory, I know it takes random chance, but in theory, doesn't that mean that 90 of the enzymes are now off? Doesn't that mean that 10 of them are still on? Those 10 that are on, what are they going to make? Tryptophan. When I have 99 tryptophans, that means I have one more enzyme that's on, and it's going to make a tryptophan. Once I get to 100 tryptophans, aren't all those enzymes off? So do I make 101 tryptophans, 102, 103, 104? No, they're all off. Okay, cool. Well, now let's say that my body uses one. My body goes over and says, I need to build a protein with this tryptophan. So it'll go and it'll take that tryptophan away and use it. So now there's 99 tryptophans. Doesn't that mean that one of my enzymes is on again? And what will it make? A tryptophan. And as soon as it makes one, won't all the enzymes be off? So won't I have 100 tryptophans? And then I'll use one. I just damaged my muscle. I need to use a tryptophan to make more muscle. Cool. So I'm going to use one. Now I've got 99. Didn't I just take away an inhibitor? So now isn't one enzyme on? So won't I make one? So don't I always have like 99 to 100 tryptophans? It's pretty steady. It seems like my cell's a genius. It seems like it knows, oh, make another tryptophan, or oh, quit making tryptophan. And it kind of does, but it's really just chemistry. If I wanted to always have 200 tryptophans around, one way I could do it is have 200 enzymes. Now, eventually you can go back and be like, well, how does your cell know to have 200 enzymes? And there's another similar thing that regulates that. But like, this is a tiny version of like, how can chemicals seem to like think, or how can chemicals like regulate themselves, sort of. All right, so that's negative feedback. I'd be pretty steady like this, like my blood sugar, like my body temperature. What would positive feedback look like? I still feedback. That's a plus. But what would be different in positive feedback? Huh? Yeah, the more I make, the faster I make more. So the faster I go. This, once I have more tryptophan, I'm kind of slowing everything down. I'm sending back signals that say slow down, slow down, slow down. With positive feedback, I say go, 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 go. So positive feedback looks more like exponential growth, something like that, kind of like parabolic growth, geometric growth, whatever math term you want to use, that. Okay, can somebody give me an example of positive feedback in a human? Negative feedback is easier. Many things I want to keep the same. I can think of 2 examples of something that once you start it, you want to go faster, 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 faster. Huh? Adrenaline, that's true. Adrenaline, most of what's causing it is I just dump adrenaline, and then that causes everything to go faster. But you're right. Like whenever I dump adrenaline in, I'd move faster. If I dumped in more into my bloodstream, then I would go faster. I don't know if adrenaline is like inhibiting things or turning them on or not, but yeah, that might be true. Mm-hmm.
I forgot what it's called. Was the thing in the cell cycle where it signals the other stages of the cell cycle? Like,
Yeah, so when you like amplify a signal,
yeah.
like once something hits something, then true. So that would be an example of positive feedback. Once I activate like one protein, that activates like 10, and each of those activate like 10 more, so now we're at 100. Each of those activate like 10 more, now we're at 1,000. That is like a positive feedback loop. I started with one signal, and that activates more, 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 more. That's a good example of positive feedback. A blood clot. Is there ever a time where I'd get a cut in my arm, and I wouldn't want to like start the clot and like get that thing clotted up as fast as I can so I don't bleed to death? Like, childbirth works this way. So there's a uterus. You're like, isn't that a balloon? And you're like, maybe. There's your unborn child. So the way that childbirth works is something like this. Let's say you are married to some guy who's named, I don't know, Alan Cotton. And he thinks he's funny sometimes. And so you're pregnant with your first and turns out probably only child. And so one day you tell him, this guy, it's, I don't know, his last name's like spelled Coton, but you say it Cotton, I don't know. And so you're like, hey, honey, I think I just had a contraction. This jerk, he's like, this is a good time to try to be funny. Because he doesn't, as my wife say, know his audience. And so normally she likes my jokes. Even if they're dumb, she's like, that's stupid, but she smiles. So she thinks idiocy is cute, +1 from me. So her husband is like, I could be a supportive guy right now. Or I could be supportive but like make a joke. And Alan's like, I'm going to make a joke. So he goes as sarcastically as possible. It's like, you guys, talking about pregnant women, you guys always talk about how difficult this is, like having contractions. You aren't even sure if you just had one. You believe that guy? Then after that, he apologizes, but of course I'm not going to accept his apology because that guy's a jerk. So anyway, he thought he was being funny, but he wasn't. Here's what happens. So when it's time to give birth to a child, this right here, the uterus, it's just a big muscular sac, and it contracts. And it contracts a little bit, and that pushes the baby's head against the opening. The opening's the cervix. And so whenever you hear them saying like the cervix is dilated 2 centimeters, 4 centimeters, 10, that just means like when the doctor looks up there, this is what they see. They see like the baby's head, literally like, they're like, the cervix is dilated this much. Why? Like, I'm touching your baby's head. And based on like, all right, I would say that's like 4 centimeters. Isn't that bizarre? Like the doctor's seeing this. So anyway, she has a contraction that puts pressure on the cervix, which tells this, hey, contract sooner next time. So your first contraction, I think I had a contraction. Butthole husband is like, and then 40 minutes later, you have a stronger contraction, 35 minutes later, 30 minutes later, 25, 20, and then eventually these contractions are just like, and you have a baby. And it's beautiful and painful all at the same time. That's positive feedback. You have a little contraction. Oh, I think I had a contraction. When you're birthing your child, there's no question you know you're having a contraction. It's just like a constant muscle spasm. So I picture like I have a giant muscle and it spasms. Sometimes I get that little spasm thing here, but now I try to envision, you know, like, I don't know, my quads. Not a spot on my quads, but like my quads just contracting as hard as they can for like 10 minutes. That sounds painful. That's positive feedback. All right. So here's the nice thing. If I look at how like gene regulation works in prokaryotes, this is like what the pictures are on your page. Most of it you already know. So if I look up there at that picture and I see a promoter. In eukaryotic cells, what's a promoter there for? Yeah, that's the beginning of a gene. At a eukaryotic cell, and I'd say something like this. I'd be like, here's my DNA, and I've got a promoter. That's the beginning of a gene. Who landed on the promoter to start reading the DNA? RNA polymerase. Does it look like RNA polymerase lands on their promoter also? Yeah. At the end of the gene, we had a termination sequence. They don't show it here, but they have a termination sequence too. The only thing that's kind of different, there's two things that are different. The only thing up here that I can see that's different is in me, I have a regulatory sequence. Over here, and that's an on-off switch. With them, they also have an on-off switch, but they call it an operator. You might remember with me, my regulatory sequence could bind a protein, and it could like turn everything off. We called it a repressor. Do you remember like seeing this picture? That was a repressor. Does it look like their operator? I know it doesn't currently fit, but does it look like maybe that repressor is close to fitting that operator? It is. So that repressor fits in the operator, but at the moment it doesn't. Okay. All these genes right here are making tryptophan. What's going to tell me to quit making tryptophan? Tryptophan. So how do you think I get this repressor to shut down tryptophan production? What's going to tell this guy to shut it down? Tryptophan. So here this is saying make tryptophan, make tryptophan, make tryptophan. And then whenever I make tryptophan, I'll see that it like goes and sticks in that repressor. Now that repressor changes shape. Now it can turn the gene off. So now, as long as that repressor is in there, my RNA polymerase like either can't land, or if it lands, it can't get past it. But it's a slightly different kind of language from us. But I start off, I've got a promoter in bacteria. I've got a termination sequence. Who lands on the promoter? RNA polymerase. Who can land on the operator? Or repressor. The only difference is I would call this a regulatory sequence in us. They call it an operator in bacteria. All right. Well, how could I get that repressor to decide to like land on there? Well, if I'm making a bunch of tryptophan, then the presence of tryptophan will cause that to fit. So again, how does a bacteria doesn't have a brain, how does it know to like control its tryptophan levels? That's how. It doesn't know. It just does. All right. So that's one way that bacteria can kind of run their genes. Trying to think if there's anything else different. The only other difference is like they call it an operon. So like in bacteria, they'll take like all the genes and just like read them all at once. If this were in a human, I'd have a gene for like enzyme A, a gene for enzyme B, a gene for that, a gene for that, a gene for that. Here they like read them all together. So in bacteria, if I was trying to like make tryptophan, the idea is in bacteria, I like either turn on all 5 genes or none of them. As a human, gene 1 would be over here, gene 2 would be over here, gene 3 would be over here, gene 4 would be over here. But in bacteria, they're all like side by side by side by side by side. I have 1 promoter, I read 5 genes, termination sequence. The logic being, if I make an enzyme 1, I'm going to need an enzyme 2 and a 3 and a 4 and a 5. If I make 50 of these, I'll need 50, 50, 50, 50. That's their logic. So they kind of like shove it all together. And so that's another difference. So if I'm looking at prokaryotes versus eukaryotes, when they cram all these genes together, they call it an operon. And then instead of a regulatory sequence, they call it an operator. All right. As far as stuff that you won't need for your test, since we're out of time here, so I've kind of got like the trip operon there. If you want to put like a little X, I wouldn't like destroy it because one day we'll learn it. But out here to the side of like the lac operon, you can like just put a little no next to it. Or as you've learned, just put a 7. Recycling joke. So I can just put a no next to that. Below that where it says positive gene regulation down here, you can skip that one. So this whole chunk right here, and then this whole lac operon chunk right there, I don't need. The one above it we did. That was the trip operon. That one's good. So this is fair game. Trip operon. So this whole box here is good. The lac operon. So like this whole box here. In that picture you can skip for now. This one down here you can skip. And then when I get to like the last page, you can skip all the last page. So all this like gene text stuff, where it says gene regulation of translation and after, and they talk about like RNAi and microarrays and stuff that's not even in the book, CRISPR, you can skip that whole page. We'll learn it when we do our next DNA technology lab, but you can skip that whole last page. That's a repeat. So like this stuff right here is still like 5' cap and poly A tail and gene splicing again. So it's fine.
Last page included gene drive.
Yeah, gene drive you can skip, yes. So I didn't realize that there was a little bit past it. So yeah, the last. I won't ask you about, because that has to do with like the lac operon. So I won't ask you that. Mm-hmm. Nope, you can skip that part. Yeah, so the 5' poly A tail splicing, you can. Yeah, kind of right there. Yep, basically like that's the last. Right there would be like the end of stuff that's fair game. And then, and then back here on prokaryotes, so those bottom 2 you can skip. Yeah, so that you can skip.
This is all fair game.
Mm-hmm.
Since we just worried about that.
Mm-hmm. Yep.